Start our morning session. I have two short announcements first. Tomorrow in the morning we will have a group photo of this conference. The exact, it should be taken somewhere during the morning session, but exact time it comes later.
I'll provide some details.
Thank you. And second, we will have a poster interchange. Those who still have their posters on the board, please put it off so that other people can put them on. Okay, and now let's start our first talk. It's from Natalia Trayanova from Hopkins, and she will tell us about integrative models of heart function in health and disease.
Good morning, everyone, and I would like to start my talk by saying I'm profoundly grateful to the organizers for inviting me here. This is one of the best modeling conferences I have been. And Nick, where are you? Thank you. Thank you so much for organizing this. I am really impressed by the program, and it's my pleasure and my honor to be here. When I wrote the abstract in this session of image-based modeling in cardiac electrophysiology and electromechanics, I decided to present to you today 4 examples of our work. However, given the time, I'm going to focus on only 2. So what I will do is I will talk about image-based modeling, and I will focus on integrative modeling of infarct-related ventricular tachycardia and its application of the understanding that we gained from that for ventricular ablation. And then I would like to spend some time talking about image-based integrative modeling of cardiac dysynchrony and synchronization. So in the first part of my talk, starting with the VT, infarct-related VT circuits, the basic science behind this research is sort of embodied in this slide. There have been numerous studies, mostly from Columbia University, Andrew Witt's lab, as well as others, that have looked at what is the substrate that underlies the formation of VT in association with an infarct scar. And they have demonstrated that the electrophysiological substrate is quite complex. And in this review paper by Stan Natel, he has put this summary graph that indicates the various electrophysiological changes that occur in the border zone or peri-infarct zone that underlie the formation of the reentrant VT that's associated with the infarct scar. So basically, it's an electrophysiological substrate and functional re-entry for a large degree. Another school of thought that started from the Netherlands, particularly Jacques de Backer, and then was followed by studies by Bill Stevenson, maintains that VT is mostly due to anatomical reentry through pathways of surviving myocardium within electrically silent scar tissue. And in this review paper by Rolf Lazara, he presented these graphs that embody the statements made by Jacques de Backer and Bill Stevenson, in which there is a tortuous propagation through these pathways that exist within the scar, and therefore the reentry occurs in this anatomical pathway. In a recent study from Johns Hopkins by Ashikaga et al., published in Circ Research, the authors for the first time looked at MRI imaging of the infarct scar, high-resolution MRI imaging, and what they noticed that in a lot of the cases, I'm not sure you can see here, the infarct was very heterogeneous. So there were regions of white, staining white appearing tissue and much more gray appearing tissue. And in addition to that, they observed both figure-of-8 reentrant circuits, which have been attributed mostly to the electrophysiological contribution of the border zone or the peri-infarct zone, as well as epicardial and endocardial breakthrough, which is more typical for the studies shown here, which refer to the human rather than the dog, as in the previous slide, which has a very extensive epicardial border zone. So what we aimed with this study is to determine the contribution of the infarct scar architecture and post myocardial infarction remodeling in the peri-infarct zone to the morphology and location of the VT reentrance circuits in the infarcted heart, and then use this insight to see whether we can optimize VT ablation associated with the infarction. So here is how we created our image-based model. In this particular case, it was a canine heart, which was 4 weeks post-MI, and it was scanned MRI, DT MRI at this resolution, and then it was interpolated. If you were watching this movie, you would see as it goes through, the region of the infarct appears in white. And what we did after that is we went through this we developed this pipeline for reconstruction of image-based models. And if you're interested in it, the paper is published in the Experimental Physiology Journal that's lying around here in the hallway, the one that Dr. Patterson promised us. Each of us gets a Nobel Prize who publishes there. Anyway, so if you're interested in learning more about our pipeline for reconstructing these models, you can refer to that paper. In essence, we use the MRI images to delineate between the medium and the myocardium, and then we used what is called the fractional anisotropy approach to delineate the region of infarction, and then we used gray level segmentation to delineate from that the regions of inexcitable scar and the peri-infarct zone, which have different intensities in the images. As you can see, when you obtain the reconstruction of the scar, it is an interdigitation, quite complex interdigitation of the scar and the zone of the peri-infarct shown here in yellow. So we expect these tortuous pathways of propagation to occur. So here is the complete model. In addition to we reconstructed the entire heart. So we have the ventricles and the atria. You can see the peri-infarct scar and actually the peri-infarct zone, the inexcitable scar. We use then the DT MRI images to reconstruct the fiber orientation in the model, and then we created the computational mesh. Now, just creating a computational mesh is a very difficult problem, and some of you who have struggled with that are probably really weary of that. We just published a paper. It's in collaboration with Gernot Planck, mostly in his group, and the original developers of this code, in which we describe all the properties of these meshes and the way you construct them. It's a very powerful meshing tool that we now use in all our studies. So we meshed that heart to create the computational mesh, and then we incorporated electrophysiological properties in the model. As you can see here, we have the atria were electrically insulated. We have a layer of non-conductive tissue separating atria from the ventricles, as shown here. The scar is an insulator, and then we had electrophysiological modifications in the peri-infarct zone to represent electrophysiological remodeling. They are based on the work by Penny Boyden and Candido Cabo, as well as others at Columbia. We have changes in the conductivity in the peri-infarct zone, as well as the expression in the ionic current. So as you can see here, the peri-infarct zone is characterized with somewhat larger action potential duration and therefore larger refractoriness. So then we induced VT in this reconstructed model. The way we did that is we followed the protocol of programmed electrical stimulation, exactly the way it is done in the hospital. So we got that from our collaborators at Hopkins. We paced that heart at 300 milliseconds several times, and then in 2 beats at decreased coupling interval. In our case, we paced endocardially from 28 sites, moving around the catheter as you will. And very similar protocol again is done in the clinic. They use less pacing sites. However, when we did this protocol, what we noticed is of the 28 sites, we induced 8 VT morphologies. All of them have epicardial reentry. And I'm going to show you movies of epicardial view, endocardial view, and transmural view. So initially, you will see the pacing. So it's been paced from site close to the apex, the pacing stopped. You can see here, if you watch this, there is propagation through the scar, through these regions of partially viable myocardium. So again, I'm playing the movie. You can see here that decreased conduction velocity and increased action potential duration within the peri-infarct zone results in conduction block and initiation of figure of 8 right here. And then if you watch the movie further, you can see the endocardium, we have a breakthrough, appears as a breakthrough. And so if you calculate, let me go back for a second. If you calculate all the phase singularities that are associated with these VTs that are plotted here, you can see, first of all, that of the 8 morphologies, VT morphologies, 6 of them were very similar. You can see the chirality is the same, the frequency is nearly the same, and then we had 2 morphologies that were different, and the chirality was different. So these are the phase singularities, and again, if we co-localize the phase singularities with the peri-infarct zone, I'm not sure how I ended up with that, but sorry about that. Okay. So if we co-localize all the phase singularities with the peri-infarct zone, you can see that they are all located on the surface within the region of the peri-infarct zone, which was very important to us because that has a very significant implications for ablation. So then it is very important to understand that the dog has a very large epicardial border zone. So in order to relate that to the human, which has a smaller epicardial border zone, what we did is we morphologically eroded the peri-infarct zone and decreased its volume by 50%. Morphological erosion is an operation by which you preserve the shape of the region, you just decrease the volume. So when we did that, this reduction resulted in the disappearance of the peri-infarct zone tissue from the surface of the heart. And then we performed again the same stimulation protocol, and of 28 sites, we induced 9 ventricular tachycardias. None of them had a figure of 8 reentrant representation on the epicardium or endocardium. All of them had breakthroughs on both epi and endo. And all of them, however, as you'll see here, this is how it appears on the surface, and you'll see as a breakthrough. However, all of them had a intramural figure of 8 morphologies. So what this tells us is that these 2 concepts, one that has been observed in the dog, where you have always epicardial figure of 8 reentry, and in the human, where most of the time you have breakthroughs, these 2 concepts are not completely different. They can be associated with the fact whether your epicardial border zone extends to the epicardium or not. Okay, so then we did the same experiment where we paced at these 28 sites, and we watched in this case of decreased epicardial border zone, what is the manifestation of the reentrant circuit. Again, these the locations of the breakthrough, that is the mean frequency of the 9 pacing locations. 6 of them had the same morphology of the reentrant circuit, and 3 shown in red were different. You can see the frequencies were higher. And again, if we co-localize these now with the peri-infarct zone, the projection of the peri-infarct zone, you can see it here in a transmural view, semi-transparent. You can see that the 3 morphologies in red are here, and the 6 morphologies are here. So they are very clearly co-localized with where the epicardial border zone would be. And then what we did is we decided to really figure out what is the role of that epicardial border zone. So we removed it completely, left only the scar, and what was previously peri-infarct zone is now normal myocardium. And then we did the same. As you can see here, we are pacing again, but in this case, we could not induce a VT in any case. And you can see here, that was a transmural movie. So that does not mean that the wavefront did not propagate through these regions around the viable scar. What we could not do is we could not induce the reentry with pace mapping as done in the clinic. So what we learned thus far is that the VT organizing centers and NIT isthmuses are located within the peri-infarct zone. VT is not inducible without electrophysiological remodeling in the peri-infarct zone, although strands of myocardium traverse the scar. The morphology of the peri-infarct zone determines whether reentrant activity is on the surface or it is transmural. So how can we learn from this and how can we apply that in our attempt to optimize the ventricular ablation procedure, which has actually about between 50 and 70% of success in the human. And here, what we did is, knowing where the phase singularities and the locations of the breakthrough are, we wanted to do an intelligent ablation. So what we did is we constructed hemispherical masks of radius 8 millimeter, which is exactly the scar that's created by ablation in the clinic. And then we constructed those, and we located them in such a way that they cover the phase singularities. You can see here we can't cover all of them. And to cover all of them, we need to perform 2 ablations adjacent to each other. So we did the first ablation. And here is the movie. This was the pre-ablation movie. Here is the ablation movie. You can see here the ablated region. And there is no longer VT induced. However, it was not always the case. Of 8 previously induced epicardial reentries pre-ablation, when we did this ablation, we were left with 2, which were no longer epicardial. They were intramural figure of 8, which tells you that we are decreasing the epicardial border zone again, and the reentry retracts, so to speak, intramurally. And then we need to do a second ablation to do that. And here is the second ablation located close to the first, and you can see here we no longer, no, this is in a case where we actually were unsuccessful. And then we performed ablation 2, in which we placed next to it the ablation site, and you can see we no longer could induce a VT. So in conclusion, in addition to the electrophysiological insight that we gained from this study, we also now have gained insight to guide ablation. So it is a 2-step process in which we understand what are the mechanisms that underlie that process, and then we use that to guide the actual clinical procedure. So this concludes the first part of my talk, and what I want to do now is move on the second application of our image-based modeling approach, in which I want to talk about electromechanics, and specifically about the loading effects in the normal and failing hearts, and what are the implications for cardiac resynchronization therapy. So the motivation for this project is that the motivation for this project, as stated here, is that pathophysiology of dysynchrony remains incompletely understood, and effects of CRT are not fully known. As you might know, the resynchronization therapy does not work in about 30% of patients, and stratification of patients who need resynchronization therapy is not very clear, and a lot of problems remain to be worked out to be able to better refer patients for this procedure. So in this talk, I focus on characterizing the relationship between the electrical and mechanical activation in the normal and failing hearts under different loading conditions. So when a myocyte is activated, an action potential is generated. Contraction follows shortly after that. The myocyte begins to shorten. There is, however, a delay between the electrical activation time and the mechanical activation time, which is termed electromechanical delay. The electromechanical delay has 2 components. One of them is intrinsic, which is a cell-specific EC coupling. And Andrew McCulloch talked about that yesterday. So the cell can generate different twitch that is associated with its intrinsic properties. In addition, the electromechanical delay has a second component, and it depends on the loading conditions, basically where the heart is spaced. So we will be focusing in this, we focused in this study on how electromechanical delay depends on the loading conditions. We did not look at the role of the intrinsic self-specific EC coupling. This is a seminal study by Princeton and co-workers in which they looked on the epicardium at the electrical activation and mechanical activation in sinus rhythm and at two pacing locations. And what the authors noticed, that electromechanical delay was very different at the base and apex. You can see here there was always a difference between base and apex. And in addition to that, that delay depended very much, and the difference as well, depended very much on the pacing site. In further studies by Ashikaga and co-workers, they embedded beats in the transmural direction and were able to record electrical and mechanical activation across the ventricular wall in one transmural location across the wall. And this is their plot from sinus rhythm, and you can see here the electrical activation and the mechanical activation in sinus rhythm. And they noticed that there was a larger electromechanical delay at the epicardium as compared to endocardium. This is sinus rhythm, so the epicardium is the region of the myocardium that activates later under normal conditions. So from these studies, it is clear this in the transmural direction, although it's only at one location, and also from the epicardial studies, as well as from studies from Andrew McCulloch's group, where they have looked in simulation results, that there is this electromechanical delay in the heart that could be distributed quite non-uniformly. So what we did is we asked the question, what is actually the three-dimensional distribution of this electromechanical delay in the normal heart? And then can we assess that distribution of electromechanical delay in the failing heart, because this could be particularly important for resynchronization therapy. And we aim to do that using our new approach, which we call image-based electromechanical modeling. So this is, as I said, we have developed that pipeline that's published in the paper that's outside, which now is, we have perfected in such a way that we can reconstruct hearts fairly quickly from imaging modalities. This shows our various reconstructions from MRI and diffusion tensor MRI. We have reconstructed a mouse heart. This is a rabbit heart from Oxford, which we have done our own reconstruction. This is an infarcted rabbit heart, which was provided to us from WashU. This is a normal canine heart, which actually we have reconstructed 5 hearts. We have the infarcted canine heart I just showed you. We have also 5 hearts with heart failure. And this is our first human reconstruction. You can see we have delineated the atria and the ventricles. They are all equipped with fiber orientation. Again, these are the papers with the reconstructions. Now, in this talk, in this electromechanical study, we use the normal canine and the heart failure canine. So it is a multi-physics, multi-scale problem, and we needed, again, to generate the meshes for our electrical and mechanical components. The electrical mesh, I talked about that, the way we use it, the approach is in our new paper. Then we had to develop our own approach to construct mechanical meshes directly from images. In both cases, the meshes are generated from directly segmented images. So we developed that, the paper we are preparing for publication. So this is our mechanical, image-based mechanical mesh. We reconstructed that. So in addition, from the diffusion tensor MRI, as before, we incorporated the fiber orientation. But as you know, in mechanics, the laminar sheets are important. So we used all the information from the diffusion tensor MRI, and we also reconstructed the laminar sheets. So that's our model from the structural point of view. In terms of multi-scale properties, we have the electrical component, in which we have an ionic model of the canine. The mechanical component, we incorporated the myofilament model of Rice and co-workers that Andrew McCulloch talked about, and it was modified for the canine by Campbell and co-workers from Andrew's lab, which we adapted for our model. So we have true multi-scale representation in both the electrical and mechanical components. We have also pseudo-Purkinje representation, and then we embedded all that model in a model of the circulatory system, which we modified also from Andrew's lab. Our modification consists in ability to maintain constant volume during isovolumic contraction. All this constitutes our multiscale electromechanical model, and we use that in the normal heart, and then we model the failing heart. So this is the mechanical mesh for the failing heart. Now, since this is an image-based reconstruction in which we have the diffusion tensor MRI, we are here. It is well known that there is a remodeling that takes place in the sheets, particularly in the failing heart. This is work from Ray Winslow and Elliot McVeigh, performed by Patrick Helm. So our failing heart incorporates this remodeling of the sheets already. In addition, we incorporated electrical changes in the failing heart. We have the change in the conduction velocities, the altered ionic currents, and altered calcium transient. We incorporated changes in the mechanical properties. We have increased in passive stiffness based on experimental data. And what you see here is the single twitch in the normal and in the failing heart. So this is our complete model of the canine heart. You can see electrical propagation here. I'm just showing the ventricles. Here is the contraction. The heart is contracting. You can see the torsion. And here is a brief movie of the strain that develops in the normal heart. So what we did is we first looked at sinus rhythm, and we constructed the isochrones of electrical activation and mechanical activation. You can see here mechanical activation is slower than the electrical activation. And then we constructed the maps of electromechanical delay. So for this here, for the first time, we have a full three-dimensional picture of the distribution of electromechanical delay in sinus rhythm in the dog heart. You can see here that there is definitely a transmural gradient, particularly close to the basal region. And this is consistent with the study that I showed you by Ashikaga, where they looked more or less in this area right here and observed that gradient. In addition, gradient exists as well in the apex to base direction. Notice around the base, we have much less transmural differences in electromechanical delay. So when we plotted the electrical activation time versus the onset of cell shortening, and you can see here for the points in the LV mid wall, we have a slope of 1.58. What that means is that the later depolarization is associated with a larger delay. In other words, regions that activate later in the sequence have a larger electromechanical delay. What is this due to? This is a phenomenon of pre-stretch, which has been proposed previously, and it consists in the fact that when the heart contracts, begins to contract in sinus rhythm, the contraction of this region, which is early activated passively, stretches the fibers that are located in the more remote regions, and therefore we have this pre-stretch. There are other reasons that lead to this pre-stretch. For instance, also the fact that during isovolumic contraction, the pressure that develops when the starts to contract from the apex, the pressure that develops here additionally causes pre-stretch of the fibers that are located in this region. So as we can see, the regions that are late activated, for instance, black, red, have a pre-stretch. So therefore, we have an electromechanical delay there because the cells who have been pre-stretched need to begin to shorten after that pre-stretch. The same phenomena occurs across the ventricular wall here and the two epi. So that's the explanation of why we get. Again, this has been suggested as a mechanism previously. However, what we did is we correlate the maximum pre-stretch, as you can see it here. So we plotted the magnitude of the maximum pre-stretch in 3 dimensions throughout the heart, and then we correlated that. So these are the regions of maximum pre-stretch, and we correlated that with the delay that occurs. And you can see here that we conducted statistical analysis, and the regions that are associated with early depolarizations, they have a less electromechanical delay versus the regions that activate later, they have a larger electromechanical delay. I want to digress here just for 1 second. When Martin Nash gave the talk yesterday, he said, whoever presents, ask them how they validated the model. And we started first as everybody else who does electromechanical studies looking at experimental data that has been published. That was the mostly papers by Chicago and co-workers. But then we did our own model validation. We collaborate with Elisa Conefago's group in Columbia University, and we have conducted extensive validation of our models with her ultrasound imaging. What Elisa does is she, these are maps of incremental strain. She images the electromechanical wave, and we, this is our first study with the rabbit, then we did the dog, and so forth. We have presented that at Heart Rhythm, and also we have a couple other abstracts published in that in a paper in preparation, but we have done extensive work with her on the validation of this model. Anyways, so far what I presented to you was the electromechanical delay in the normal heart during the sinus rhythm, very small portion of what we are interested in. So what I want to talk to you about the conditions that are different from normal, which is left bundle branch block and biventricular pacing that applies to CRT. So here again the electrical activation maps. I've shown you this before, and now I'm adding maps for LBBB and biventricular pacing, which you see here. In LBBB, you have a late activation of the left ventricular wall, which is corrected by this biventricular pacing here, the movies of electrical activation that correspond to these maps. You can see the late activation here and correction by the biventricular pacing. And here are the electrical activation maps of the failing heart. So again, this is a sinus rhythm, and we have LBBB and biventricular pacing. The electrical activation movies, again, activation is much slower in the failing heart. You can see again the late activation of the wall and the biventricular pacing. And then I'm showing here the fiber strain. You have seen this movie before. Here is the fiber strain in bundle branch block, the late activation. You can see the red region right here. And then this is again corrected. When you compare these movies to failing heart, you can see how much weaker contraction in the failing heart is. We still have the large fiber strain in the left ventricular wall here. And here are the fiber strain traces. They have a much smaller magnitude here. These are shown in transmural direction. Again, here is this is what I showed you before. These are the new strain traces that we calculated. And here are the electromechanical delay maps. Again, this is, I put it here just for reference. I've showed you that before. And here we have the electromechanical delay maps, again, in the normal heart for these 2 cases, and in the failing heart for these 3 cases, sinus rhythm, bundle branch block, and biventricular pacing. Now you can see that they are very large, they are larger transmural delays that occur in the failing heart as compared to the normal heart. In addition to that, we have a larger delays in the LV free wall. And this exists pretty much throughout from apex to base. And again, we did the same study. We correlated the magnitude of the maximum pre-stretch in all these cases. And you can see that in all cases, in the normal and in the failing heart, there was a difference between early activated regions and late activated regions. In the early activated regions, we have a much smaller electromechanical delay. And in the late activated regions, we have a larger electromechanical delay. And again, it was consistent across pacing protocols, in other words, across loading conditions, and both in the normal and the failing heart. So it appeared to be a universal phenomenon. And then we plotted the same plot, the electrical activation time versus mechanical activation time. This is what I've shown you before, and these are the new results. In addition to that, and you can see in all cases that slope is larger than 1, again indicating that late activated regions are associated with a larger electromechanical delay. Now notice that in this case, we have a pretty significant difference between the normal heart and the failing heart. In the failing heart, we have always these slopes are larger. So in heart failure, the late activated regions are associated with extended electromechanical delay that extended additionally as compared to the normal heart. So this was an important conclusion we drew from here, which can be very important in understanding how to improve CRT. So in our hands, we have been able to develop, in conclusion to this talk, in our hands, we have been able to develop these image-based electromechanical models of the normal and failing hearts from these high-resolution MRI and diffusion tensor MRI scans. Again, it's a very extensive pipeline that we have developed, and we have tested numerous applications in the development of that. You can find the information in the paper presented outside. It's a very powerful tool because it allows you to reconstruct any scanned heart, and our hope is that we will be able to use this approach in the clinic to reconstruct images in patients and use our simulation tools to also apply to situations that are valid in the clinic. In this particular study, we also demonstrated that there is a three-dimensional distribution of electromechanical delay that is non-uniform and depends on the loading conditions. In both normal and failing hearts, we found that regions that depolarize later in the activation sequence are always characterized with longer electromechanical delay. And there is a correlation between the three-dimensional distribution of electromechanical delay and the local magnitude of myofiber pre-stretch. And then the increase in electromechanical delay, electrical activation time, is larger in the failing heart as compared to the normal heart. And I would like to conclude my talk with acknowledging all the people who have contributed to this talk, the students who have worked on that, Jason Constantino, whose PhD is on electromechanical modeling, CRT, and so forth, and Hermenegilda Revelo, who works on VT and ablation, and my postdoc, Slava Gurev, who developed all the electromechanical modeling, and Fijoy, who developed the reconstruction pipeline for our image-based modeling. The programmers in my lab, Rob Blake, who is an invaluable contributor to all of the studies, Ted Lee, who generated the mechanical meshes, Gernot and Ed, with whom I have a long-standing collaboration, who contributed tremendously to the technical aspects of my work always, Jeremy Rice, for working with us on incorporating his myofilament model, Pat Helm, who scanned for us some of the hearts, particularly the heart with infarction, who was a postdoc in Virginia, ran to NIH to scan the heart, and Elisa Kunofagu, with whom I work on the validation of our electromechanical models, and most importantly, the people without whom all this would have been impossible, the cardiology, the people in the JHU cardiology, Henry Helprin, with whom I work on VT ablation, and David Kass and Al Lardo, with whom I work on electromechanical modeling and resynchronization therapy. And I would like to acknowledge NIH, NSF, and AHA for supporting my research. Thank you very much.
Thank you very much for excellent presentation. Okay, Peter.
Natalia, a beautiful talk as always. But what orthotropic are you using an orthotropic constitutive law for your mechanics? And are you basing it on the fiber sheet architecture?
Yes.
And how do you modify it for the failing?
How do we modify it for?
Failing.
So what we do is.
Please repeat the question.
Oh, sorry. Peter is asking whether we model our failing heart as orthotropic, and right?
Mechanically.
Mechanically, yes, and whether our constitutive laws are orthotropic, and my answer to that is yes, they are. And the second part of the question was?
The question is really, how do you modify your orthotropic constitutive law to account for the changes that occur in the failing?
What we do is, I believe that Slava did that on the values that are available in the literature. What is interesting here is in our diffusion tensor, we do not when we map the diffusion tensor onto our heart, we do not map vectors. He interpolates the tensor directly. So that is the approach that he has taken, and in this way he incorporates these orthotropic properties. So we do not, we interpolate tensors throughout.
That'll give you anisotropy, but it won't give you mechanical properties.
Right, but some of the constitutive the properties are modified based on the data we found in the literature, as I showed in the heart failure slide. Does that answer your question?
No, but we'll talk later.
Okay.
I'm sorry, Andrew was.
That was fantastic, Natalia. I'm trying to remember, but when we did one of those first electromechanical models in a normal heart with pacing, we saw negative electromechanical delay. It was possible to get negative electromechanical delays.
I'm aware of that.
And I was looking at the plots, the scale of the plots didn't have them, but some of your scatter plots looked like they might have had negative. Did you see that?
No, we didn't. I'm very well aware. We looked at your paper. We pored over that. You know, I remember your plot even, which goes down here. You have the negative. We never saw that. I mean, no, we never saw that.
I wonder why. I don't know that experimentally it's been seen, so.
Yeah.
Natalia, really, really lovely. The two questions, really quick ones relating to the infarct water zone. And the first one is to go back to the question of validation. Do you or do you intend to, and I'm sure the answer is yes, have you or do you intend to compare the model predictions with electrical mapping studies?
Yeah, this is it is of course a very good question. So what we have done is, it is a little difficult when the heart is first scanned before they did and they didn't do a mapping on it. So we have numerous hearts in which we have SOC mapping. So it's done in Henry Halperin's lab. They have SOC electrode mapping on the epicardial surface, and then they have an MRI. They didn't have the DT MRI. So we had to develop specific procedures to map fibers from one heart in the other, and we have done validation in these hearts. This one that I showed you, I do not have experimental electrophysiological recordings, but we do have in other hearts for which we did not have diffusion tensor MRI exactly. So the fibers were mapped differently. Okay, so we just got a grant funded actually recently, and there is a whole slew of studies that we are planning to do there using the same. Actually, we'll do it in pigs, and we have a protocol now in which you do first the electrical mapping with the SOC, and then you excise the heart, and then you do ex vivo imaging of diffusion tensor over MRI, and then you compare. And it works very well because you can compare. We'll have a second set of studies in which they do the ablation. So we will excise the ablated heart, and then we compare it as well. And then you can see that whether the prediction of the ablation coincides with what they found in the experiment based on the SOC electrode recordings.
The second question was just application of the optimization or ablation, and just any comments which you've got, and presumably these comments also from cardiology, on the application of these ideas, given that in humans your resolution for gadolinium imaging is going to be actually 1 millimeter, and your electrical mapping is.
Excellent question. So we have tackled that question, and I have actually a backup slide of that. This is So we have. Let me just show you that very briefly. So this is. Okay. So this is a heart that we got from the clinic. And it's an attempt to do, this is gadolinium enhancement. It's an attempt to actually reconstruct some of that. And it's very hard because it's completely different from this high resolution ex vivo imaging. So we attempted to do that via applying numerous image processing techniques to get the infarct scar much more precisely outlined. It is tough. We are working on it. And my hope is that we actually will be able to apply that.
Texture analysis might actually give you some part of the way.
Right. So, yeah, we are.
Yes, Major Plumter. Let's switch computers, and last question from Alan. Yeah.
Quick question, need a take-home message on the VT strategy for the clinic. So, of course, as you know, what they do right now is they do pace mapping, and they try to find the circuit, and then they ablate the circuit. Yeah. What strategy emerges from your modeling that I can tell our docs how to do it?
Yeah, so if you have seen some of the posters at Heart Rhythm, so Henry Halperin's lab had a a poster on swine hearts, and that's how it's related to the clinic. So they believed, initially, everybody believed that you need to go and target in the scar these pathways, right? That's the standard goal. You target the pathway and you eliminate. We demonstrate that that's not that. You need to find a region of functional remodeling that actually helps maintain these circuits, as I demonstrated. If you do not have that functional remodeling, you don't start the circuit. So in communicating with Henry Halperin, we convinced them to try to ablate in these pigs, not areas that look really white, which is the scar where initially it was believed that possibly some of these tortuous pathway lie, but side regions where you have a more gray-looking areas, which we believe are the peri-infarct. And in the poster that I presented at Heart Rhythm, all their ablations in those kinds of, they call them heterogeneous gray regions in their poster, that's where they succeeded the most. So it's based on this analysis.
Okay, I think we need to stop discussion and go to